record the talk, and I welcome people to post those questions in the chat. I will also ask you to please silence yourselves so that there's no noise in the background for Yvette's presentation. And then at the end, we'll have a couple of minutes for people to chime in and ask questions. So Yvette, thank you so much for your presentation today, and time is now yours.
Yep. Thanks, Diane. Thank you, Diane, and thank you to the CIHD organizers for inviting me to be part of the seminar series. I visited Utah a couple years ago as part of the Rising Stars workshop, and without exaggeration, Salt Lake City is the most beautiful place I have ever visited. So I really hope that everyone gets vaccinated and I can come visit our friends and collaborators in Utah again, as well as to enjoy the sight of these inspiring mountains. Just a little bit about my lab. My lab is interested in mitochondrial heme metabolism. And today I'm going to, yep.
I'm going to interrupt you. Your presentation is showing up as presenter's view.
Oh.
That's it.
Wait, okay. Sorry, I just need to see whether I can swap that so that I can see my notes. And now my computer froze. What's going on? My apologies, this is Zoom.
About this, if it's frozen, you need to just unshare and then reshare again in the new sort of status.
Now it says my internet connection is unstable.
I don't know what that means. Okay, I don't see, right now, yep, now I see your presenter's view.
Okay.
I think there's a way, yeah, that's not a presenter's view. Sorry, we should have worked this out before.
Like, it went black again.
Hey, Diane.
Yes.
It would be nice if the secretary would send a calendar invite instead of having to sort through all my email to find the link.
I can do that. We can do that for the rest of them. That's easy to do. Thanks for the suggestion. How you doing, Yvette?
Sorry, my computer is like making whiny noises and working hard. Okay, is that?
Yep.
How is that now?
That's great.
Okay. So I really hope everyone gets vaccinated and we can all get back to normal again. So my lab is interested in mitochondrial heme metabolism, and today I'm going to talk about some of the things that we've worked on in the last 3.5 years or so. Time flies when you're having fun. So I don't really need to tell this audience what heme is, but I'm just going to give a really quick reminder of what this wonderful molecule is about. So heme consists of a central iron. So this is in blue here, that is chelated by a porphyrin ring. And it functions as the prosthetic group of hemoproteins and plays a wide range of roles in cells, ranging from things like oxygen transport in hemoglobin, as well as regulation of circadian rhythm and mitochondrial respiration as your OXPHOS cytochromes. So regulation of heme synthesis in the erythroid cell is very complex, and the heme tetrapyrrole ring is synthesized via a series of well-characterized enzymatic steps, which begin and end in the mitochondria. So in the first step, glycine is imported into the mitochondrial matrix and is used to make ALA via the enzyme ALA synthase, which is tightly regulated by the mitochondrial protein unfoldase, ClipX, which I'm going to discuss. So this is the committed step of the heme synthesis pathway. And then ALA is then exported into the cytosol via some transporter that we have not yet identified, where it then undergoes a series of enzymatic reactions, ending with coproporphyrinogen, which is then transported into the mitochondrial intermembrane space, where it is then oxidized to form protoporphyrinogen 9. And then protoporphyrinogen 9 is imported into the mitochondrial matrix via a process that requires a protein called TMEM14C, and then is oxidized by PPOX to form protoporphyrin 9. And then the other part of heme synthesis requires the import of iron into the mitochondrial matrix so that ferrochelatase can then metallate protoporphyrin 9 to form heme. And then finally, heme is exported into the cytosol to make hemoproteins. So a lot of the studies in the field have focused on the first and committed step, ALA synthesis, as well as the last step, which is mitochondrial iron import. So we don't really know as much about regulation of the intermediate steps, which is also pretty important because you need your intermediate steps to keep pace with heme synthesis if you are going to upregulate your first steps and your last steps. So that's something that we are going to talk about today. And when we think about iron and heme, many of us think about red cells. So during terminal erythropoiesis, developing red cells use about 90% of the body's iron and heme to make heme and hemoglobin, which then transports oxygen to all tissues in the body. And therefore, for our purposes, the red cell is an ideal model system to study iron and heme, as their predominant function is to synthesize heme and globin. So there are two major gaps in the field, which I have listed here. While the core proteins of iron metabolism and heme synthesis have been well identified, we don't really fully understand how cells couple heme synthesis to the heme and iron needs of the cell. And we definitely do not fully understand how erythroid cells developmentally regulate iron metabolism pathways so that red cells can handle this much iron and heme during development. So what are the molecules that regulate heme synthesis during erythroid development? How do red cells coordinate heme synthesis and iron uptake without accumulation of toxic intermediates while ensuring an adequate supply of iron and heme for erythroid cell function? And then, how does erythroid differentiation regulate heme synthesis? The other major question in the field is really the converse. We know that erythroid differentiation causes this massive upregulation of heme synthesis and iron uptake for hemoglobin synthesis. But beyond this, beyond hemoglobin production, a lot of things happen during terminal erythropoiesis, including cell cycle progression, nuclear condensation, and enucleation. And iron is required for the correct progression of these processes. At this point, we still don't really understand how iron functions as an effector of terminal erythroid differentiation. So that's also a question that we are very actively interested in. So iron and heme disorders are most commonly associated with hematologic disorders because red cells synthesize the vast majority of heme in the body and also require large amounts of iron for their function. And these diseases are a major public health issue because 20 to 25% of the population suffers from some form of iron deficiency anemia. And most notably, these sufferers are women and children. some proportion of these anemias are caused by genetic defects, and there are also many modifier genes that are yet to be discovered. There are also some clinical studies in Europe that show that oral iron supplementation is really not that effective in treating this problem, showing that we have a long way to go in dealing with this problem of iron deficiency anemia. Defects in heme synthesis also cause accumulation of photosensitive porphyrins, which also results in porphyria. So this is a rare disease. And lastly, the body does not have a mechanism to excrete iron in a regulated manner. And therefore, inability of the body to correctly regulate iron uptake and utilization can also cause iron overload. This causes iron accumulation in organs such as the heart and liver, which can lead to tissue damage. So in the US, hereditary hemochromatosis affects about 1 in 200 to 500 individuals, which is quite a lot of people. The takeaway message here is that poor understanding of the molecules that are responsible for iron and heme metabolism poses a quite formidable barrier to understanding these diseases. So within the major questions of the field, here are the goals of my lab. First of all, we are interested in understanding how individual cell types adapt iron metabolism for their specific needs. And then to use this information to understand disorders of iron metabolism and to design therapies for these diseases. And lastly, we are trying to understand how iron metabolism regulates terminal erythropoiesis. So today, so with the goal of understanding how something as ubiquitous as heme synthesis is regulated erythroid cells, I'm going to tell you 3 short stories about our work in this area. So the work began when I was a postdoc collaborating on a project to understand the role of a ubiquitous housekeeping protein unfoldase and chaperone, ClipX, in yeast heme regulation. So shortly after we discovered that ClipX was required for heme regulation in eukaryotes, our collaborators in France, led by Hervé Puy, came to Barry saying that they found a mutation in this ClipX gene that co-segregated with porphyria in a family. So, and they found, using whole exome sequencing, they found that all the heme synthesis genes were normal. So today I'm going to tell you about what we learned about ClipX and heme from looking up the patients and how we started thinking about iron supplementation as a potential therapy for this porphyria. And then after I started my lab, I wanted to more fully understand ClipX and its role in heme regulation in mammalian cells. So the second part of my talk is new data, and we'll tell you that we basically found that firstly, not all eukaryotic heme synthesis is the same. The function of ClipX in heme synthesis was really not that conserved in yeast and vertebrates. But we also found that ClipX regulates basically all the steps of heme synthesis that takes place in the mitochondria. And lastly, if there is time, I'm going to tell you about a side project that will hopefully become a more central project in the lab, and how we used zebrafish iron mutants to understand how iron transport can regulate erythropoiesis, and to show that just iron availability is not enough, and you need the presence of these iron transporters for iron to properly regulate terminal erythroid differentiation. So I'm going to start my talk by giving a very brief introduction to EPP, which is one of the most devastating diseases caused by a heme disorder. So EPP is, the longer name is erythropoietic protoporphyria, and this causes painful skin blistering and tissue damage. And the most common cause of EPP is FEC deficiency, which impairs heme synthesis and allows protoporphyrin IX accumulation. There are also variants of this disease, such as X-linked protoporphyria. So this is a C-terminal gain of function deletion in ALAS2, which results in excess protoporphyrin 9 accumulation downstream. So here we don't have a heme defect because there is an excess production of upstream reactants rather than a defect in heme synthesis, like in FEC mutants. There are also EPPs of unknown etiology, and this comprises of about 5% of porphyria patients in the US. So back in 2017, our collaborators, Hervé Hui and Gail Nicholas at the U of Paris had an EPP patient that had vastly elevated total porphyrin and erythroid protoporphyrin, as well as free protoporphyrin 9, while their FEC activity was pretty much decreased. Whole exome sequencing eliminated the usual suspects, FEC and ALAS2, as well as some not so usual suspects. And they found that the proband's father and uncle also had protoporphyrin 9 accumulation, while their FEC, ALAS2, and iron metabolism genes were normal. And this disease had a dominant inheritance pattern. And whole exome sequencing established that all of these patients had a mutation in the ClpX gene that co-segregated with the disease. So what is ClpX? We don't really hear about this in graduate school when we take our classes in protein degradation in animal cells or any kind of animal cell biochemistry. And the reason for this is that most of what we know about ClpX is from studies of bacterial ClpX within the context of the prokaryotic ClpXP protease. So ClpX is an ATPase that functions as a hexameric unfoldase in complex with ClpP to form the ClpXP protease. And ClpX recognizes unstructured protein tags and then globally unfolds their tertiary structures in an ATPase-dependent manner. And then in a second ATPase-dependent step, ClpX then feeds these proteins into ClpP for degradation. So this is the main protein quality control mechanism in bacteria. So this process was so prokaryotic focused to that point. In 2012, we really didn't know of any independent functions of ClpX and ClpP. At that point, nobody really cared about eukaryotic ClpXP because the ubiquitin proteasome pathway takes center stage in eukaryotic protein quality control. However, more recently, in 2016, it was shown that ALAS was one of the ClpXP substrates. The other known function of ClpX shown in yeast in 2015 was that ALAS in yeast, ClpX helps to unfold ALAS to incorporate its cofactor PLP, which is the active form of vitamin B6, so that ALAS can function in heme synthesis. We also showed that in zebrafish, knockdown of ClpX causes anemia due to heme deficiency. So going back to the patient mutation, the disease mutation was in the well-conserved Walker A domain, which is part of the ClpX ATP binding cleft. Since ATPase activity is central to ClpX function, Tanya Baker's group assayed ATPase activity of both the wild type and the mutant, and found that the mutant has no ATPase activity. And they then mixed the wild type and the mutant, and they found that the mutant protein suppresses ClpX wild type ATPase activity, suggesting that it can function as a dominant mutant in a ClpX complex, which is quite consistent with how the disease was inherited in a dominant manner. We then wanted to see if the mutant ClipX was causative for the erythroid protoporphyria. So we did that by expressing either wild type or mutant ClipX in mouse erythroid leukemia cells. And the first thing that we observed was that in the mutant expressing cells, the protoporphyrin 9 levels were much higher than the wild type expressing cells. And since ALAS is a ClipX target, we examined ALAS activity and found that in the mutant expressing cells, ALAS activity was significantly increased. We then probed for the ALAS2 protein, and we found that it was increased in the mutant. So from these data, we found that expression of the ClipX mutant increased ALAS protein expression, which then led to an increase in ALAS activity. And we then we postulated that this consequently led to increased production and accumulation of protoporphyrin 9. We also went back and looked at ALAS levels in patient cells. So this panel on the left are lymphoblastoid cells that were obtained from the proband, as well as the proband's unaffected brother and the other patients which had a ClipX mutant. And what we found was that the patients had a marked increase in ALAS1 levels. We also differentiated primary CD34 positive cells from patients and the controls into the erythroid cells, and then looked at ALAS2, and the proband had elevated ALAS2 protein levels. So all these studies were done in France by Hervé's group.
So Yvette,
yes?
I have a question for you. So
Yep.
if ClipX was originally in 2015 shown to be important to get the cofactor in, so now when you overexpress the mutant, that shouldn't allow ALAS to get its cofactor, you're getting higher catalytic activity. So that implies that there's not a strict dependency of ClipX on ALS getting its cofactor.
Yes, and actually you predicted the second part of my talk, which actually shows that in mammals, ClipX is not required for ALAS activity. Although we haven't formally shown that, we haven't formally measured PLP incorporation. We did show that in the knockout. So we knocked out ClipX in male cells, and we found that actually an increased ALAS activity and protoporphyrin 9 production. So, I think, we have shown that ClipX is not required for ALAS activity in vertebrate cells.
But the other, I guess, interpretation is the ATPase activity is not required for PLP binding.
Correct.
So it's the mutant can still function as a chaperone in helping ALS get its cofactor.
We're not even sure if the ClipX is required for ALAS to get its cofactor now, because the ALAS is perfectly functional in the knockout, in the ClipX knockout.
Wow.
Yeah. So yeast and vertebrates are not always the same thing.
No, I know, but Tanya Baker's 2015 paper kind of led us to believe that ClipX was essential.
Yes, which was why we did the experiment in vertebrate cells, because I think she had that paper had speculated, or it had hypothesized that the heme deficiency in the zebrafish morphins was due to a lack of ALAS activity, but this was not directly tested in vertebrate cells, which was why we went and actually did the experiment by knocking it out in male cells. Have I answered your question, or yes, no?
Oh yeah. Thank you very kindly.
Okay. All right. Yeah, I can't see people's faces now. Right. So Yes, but Dennis did predict the second part of my presentation, which I'm going to be talking about, because all these data, like, it did raise questions in my mind, which was why we decided to test out these hypotheses. But at this point, since the expression of the mutant increased ALAS protein levels, we wanted to determine if it altered ALAS turnover, since this was predicted by Kubota's group in more recently in 2016. So we expressed ClipX as well as the mutant in cell lines, followed by cyclohexamide treatment to block translation, and then quantified ALAS at several time points post-treatment using Western. And in non-erythroid cells, we found that the ClipX mutant significantly increases ALAS1 stability. And this was also true in erythroid cells, where the mutant increases ALAS2 stability. So our data at that point, so this was in 2017, led to the following models. So at that point, we hypothesized that ALAS is regulated by ClipX via two mechanisms. Firstly, that the ClipX P complex normally degrades ALAS, but at the same time, ClipX was thought to activate ALAS by incorporation of its cofactor. So in the absence of ClipX, which had not been formally tested at that time, we predicted that ALAS would not be degraded. However, also that ALAS would not be activated. So we thought that this would cause a heme defect and anemia, which we did see in our zebrafish morphins. So in our patient, the ClipX mutant dominantly suppresses the activity of the protease. So this caused ALAS accumulation. At the same time, we found that that ALAS was also being activated, and we hypothesized that this was possibly because ClipX was still partially functional. It wasn't totally dead in the heterozygote. So we predicted that this was why we saw an increase in ALA formation, which then led to protoporphyrin IX accumulation and porphyria. And this data is actually clinically significant because EPP is characterized by protoporphyrin IX accumulation, which can be caused by ALAS2 gain of function or FEC loss of function. And in FEC deficiency, we do see a loss of heme, sometimes causing microcytic anemia. But in the ALAS2 gain of function, we see protoporphyrin IX accumulation, but no heme deficiency. And this protoporphyrin IX accumulation can sometimes be alleviated by iron supplementation. So in our new EPP patient, the ClipX mutation activates ALAS, causing a protoporphyrin IX accumulation. This patient was normocytic, and the porphyria was actually more similar to the ALAS2 gain of function. So we at that time we thought, this might be a new subtype of EPP that can be treated by iron. So we decided to test this hypothesis in vitro by looking at what would happen if the ClipX mutant overexpressing male cells were treated by supplemental iron. As expected, the ClipX mutant expressing male cells accumulated protoporphyrin IX, but then treating these cells with iron citrate was able to ameliorate the protoporphyrin IX accumulation and might be a potential therapy for such patients. So these experiments were done in Barry's lab shortly before he passed away, and independently of this really interesting finding that was done in in Hervey's clinical center, who tried this out on the patient. So the proband, in addition to her porphyria, also had iron deficiency anemia. He treated her with iron supplementation, and these data are shown in red. And what they observed was that after treatment with iron supplementation, there was a marked decrease in her total and erythroid protoporphyrin levels. They also saw an increase in zinc protoporphyrin. And the patient's hemoglobin levels, serum iron, and ferritin all normalized. Transferrin saturation and soluble transferrin receptor also went up. So all these data showed two things. First, that iron supplementation did normalize the patient's iron levels, and also iron supplementation reversed the patient's EPP by bringing down free protoporphyrin 9 levels, and also slightly increased FAC activity, although her FAC activity was already pretty normal. I also want to say that getting this data from Hervé was probably one of the best moments of my scientific career, because the patient had been affected for most of her life by photosensitivity. And after she was treated by iron, the photosensitivity and her quality of life improved by a lot. And it's really not every day that we can, do something to improve somebody's quality of life. So that was really nice. So I just want to summarize this part of my talk. We found that ClipX regulates ALAS activity by control of its turnover and activation. And the ClipX patient mutation disrupts the ability of ClipX to degrade ALAS, and resulted stabilization of ALAS caused protoporphyrin 9 accumulation and a novel form of EPP. So this was significant because this was the first known protein chaperone mutation to cause EPP, and also clinically significant because it's more similar to X-linked EPP than the more common EPP in response to iron. And also, and lastly, supplemental iron might also rescue porphyrin accumulation that was exacerbated by a potential iron deficiency. So in short, these data were clinically significant because they suggest a potential therapy for future such patients.
So Yvette,
yep.
Your conclusion there, go back to the last slide.
Mm-hmm.
So that's a little different than you said in the patient that there was increased synthesis of ALAS.
Yes.
So here in your summary, it's the mutant patient just has an inability to degrade, so the protein, the enzyme accumulates. So is there increased synthesis in the patient?
So there's increased ALA synthesis, but not ALAS. We didn't check translation of ALAS. We only looked at degradation.
Okay.
Yep. So we wanted to understand the therapeutic mechanism of how iron rescued this porphyrin accumulation. So we went back to thinking about how ClipX regulates heme synthesis. So up to 2016, the Cardone and Kubota papers indicated the following models of heme regulation of ClipX, i.e. that ALAS is regulated by ClipX via two mechanisms. Firstly, that ClipX-P normally degrades ALAS, while ClipX was also predicted to activate ALAS by incorporation of its cofactor. So in the absence of ClipX, ALAS was not degraded, as was shown by the Kubota paper. However, the prediction was that ALAS is not activated. So we thought that this caused the heme defect and the anemia, which we observed in our zebrafish morpholino. So to test this model, one of the first experiments in our lab was that we knocked out ClipX and ClipP in murine erythroid leukemia cells, which are a nice model to study the biochemistry of erythroid differentiation because you can induce them to differentiate and upregulate heme synthesis by adding 1.5% DMSO. I then stained with benzidine for heme, which then shows up in brown. And as expected in our model, the ClipX null cells were heme deficient, while the ClipP null cells contain more heme than the wild type. To see if this was also, if ClipX was also required for heme synthesis in vivo, we benzidine stained the ClipX-B mutant embryos that we obtained from Zerk. And consistent with the cell culture data, the ClipX-B mutant fish had decreased heme synthesis at 48 hours post-fertilization. We then asked, is this anemia caused by a heme, by some kind of developmental defect, by failure to make red cells? So on this left panel, we looked at erythroid cells, which are marked by GFP in zebrafish. And we found that the ClipX mutants did not have any decrease in erythroid cell number. So the heme deficiency was not caused by a defect in erythroid specification. However, GAMSA staining of the sorted erythroid cells showed that these cells are morphologically kind of messed up. There is a huge variance in nuclear to cytoplasmic ratio in contrast to the wild-type cells. And these mutant cells also had ruffled cell membranes and a defect in nuclear condensation, which is characteristic of erythroid differentiation. So at this point, we were trying to figure out the mechanism. So are we observing any changes in ALAS stability in the ClipX knockout? The first thing that we observed was that there was an accumulation of ALAS in both the ClipX null and the ClipP null cells. So these are indicated in the blue and red lines. Similar to what we did in our paper, we treated these wild-type ClipX null and ClipP null cells with cyclohexamide to block translation, and then did Western blot for ALAS2 to look at the stability. So in undifferentiated male cells, ALAS2 has a half-life of about 4 to 6 hours. And in both the ClipX and ClipP knockouts, we saw a marked increase in ALAS stability. And we also saw a similar increase in stability in differentiating male cells. So this was consistent with the previous observations that ClipX-P regulates ALAS stability in animal cells. And then to determine the cause of the heme defect, we assayed ALAS activity in the wild-type ClipX null and ClipP null non-differentiating male cells. So surprisingly, and as Dennis pointed out, this is really divergent from the yeast data. Both ClipX null and ClipP null cells had an increase in ALAS activity, suggesting that ClipX, in contrast to yeast, was not required for ALAS activation in mouse cells. And this translated to increases in production of ALA in these cell lines, as well as downstream protoporphyrin 9. So collectively, these surprising data show that in mammalian cells, the ClipX-P complex regulates the stability of ALAS, but it is not required for activation of ALAS, unlike in yeast. So, since the heme defect in ClipX null cells was not caused by an ALAS defect, what is the problem?
Just want to interrupt at this point for one question from Stefano Rivella. Would you expect that overexpression of ALAS2 would look like a ClipX null in the male cells?
That's a good question. At this point, my guess is yes. I think so.
Okay. Yep. Thanks so much.
And in these cells, did you look at the levels of other intermediates? So we only looked at ALA and protoporphyrin 9. We have not looked at the other intermediates. Yeah.
Thanks for the question, Amy.
Yep.
Okay, let's go on.
Are there any other questions that I missed out on?
Not right now.
Okay. Okay, so since ClipX was localized to the mitochondrial matrix, we considered the possibility that ClipX also regulated, other mitochondrial steps in the heme synthesis pathway that were co-localized. So this work was done by Hector at CIHD. We assayed the activities of the terminal heme synthesis enzymes, PPOX and FEC, which are matrix localized. And here we found that PPOX and FEC activities were decreased in the ClipX null cells, which can account for the anemia and also for the porphyrin accumulation. We wanted to, yep.
Sorry, one more question from Ole, which
Yes.
asks, have you tried the opposite to overactivate ClipP using imipridone treatment?
No, we have not tried this experiment. This was on the list, but I think it got overlooked. But this is a good experiment to try. Yes.
Thank you.
Yep. Let's see. So yes, ClipX does regulate the terminal enzymes in the heme synthesis pathway.
So
The other thing,
concerning that regulation, does it change the protein level if you blotted for FEC in the null?
Yes, that's an excellent question. It did not change the protein levels of PPOX, but yes, it did. FEC protein levels and RNA were decreased in both the ClipX and ClipP nulls. Yep. Okay. We also wanted to test if ClipX regulates mitochondrial iron utilization because there were a couple of examples of this in bacteria. So to do this, we treated ClipX null cells with iron, and this was that was chelated to a lipid-soluble compound called hinokitiol, which is a natural compound found in the wood of trees. So hinokitiol forms a bidentate complex with iron and enables iron to bypass membranes because of its lipophilic structure. So basically, if ClipX null cells were iron deficient, addition of iron hinokitiol should chemically complement the iron defects in ClipX null cells. So in wild-type cells, iron can cross membranes through iron transporters. And when there is a defect in iron transporters, a concentration gradient builds up on one side of the membrane because iron cannot cross the plasma membrane since it's ionic. And addition of hinokitiol allows iron to bypass membranes and then be transported down the concentration gradient and go to where it is needed to make heme iron sulfur clusters or iron binding proteins. So to confirm the requirement for ClipX in iron transport, iron utilization, we treated our wild type and knockout cells with iron hinokitiol to determine if iron could chemically complement the ClipX heme defect. And we found that iron supplementation partially rescued heme synthesis in the ClipX null cells close to wild type levels. And we were also able to partially rescue heme synthesis in the ClipX B fish, the ClipX B mutant fish with iron hinokitiol. So collectively, these data suggest that the heme defect in the ClipX null cells was partially caused by a deficiency in mitochondrial iron metabolism. However, since iron was able to partially rescue ClipX deficiency, we then wanted to ask if ClipX was required for a mitochondrial iron import. So to measure this, we we carried out ICP-MS of isolated mitochondria at the University of Utah. And these experiments were done by Laurie. And we found that the ClipX null mitochondria were actually iron replete. So we concluded that the problem was likely to do with iron utilization rather than mitochondrial iron transport. But exactly how is still an open question. So, circling back to the index patient that inspired these studies, we found that iron supplementation normalized the patient's iron levels, but also reversed the patient's EPP by bringing down free protoporphyrin IX levels. So according to our revised model, these symptoms might happen because defective ClipX might cause a problem with mitochondrial iron. So iron supplementation might provide FEC or do something else with iron sulfur clusters. Might provide FEC with iron sulfur clusters that are needed to function. So I mean that's one of our working hypotheses. It might also provide iron as a FEC substrate for heme synthesis. So these studies are being carried out in our lab and are likely to require a revision in our interpretation of the patient data. So, yep.
We have one other question in the chat, which is there any defect in transferrin receptor synthesis or iron intake in your ClipX mutants or hepcidin synthesis? So it's kind of
so we have not checked. These are good questions. So we wanted to look at these in the knockout mice. However, because these are better, the mouse is a better system to look at these questions that you're asking. However, the knockout mouse is actually pre-gastrulation lethal, which is very sad. But it shows that ClipX is a very important protein. So actually we are making the conditional knockout at the University of Utah. So stay tuned. I think we might have an answer in a couple of years, but not now. Did we also try FAC and deoxyhinokitiol? Yes, yes, we did. And I think that failed to rescue. Yep.
Thanks.
Yep. So in summary, ClipX, but not ClipP, is required for heme synthesis in erythroid cells. And unlike in yeast, mouse ClipX is not required for ALAS activity. We also confirmed that ClipXP regulates ALAS turnover in erythroid cells and found that ClipX regulates the terminal steps of heme synthesis and mitochondrial iron transport in differentiating erythroid cells. And lastly, a decrease in mitochondrial iron might exacerbate ClipX EPP, and iron supplementation may decrease both mitochondrial iron deficiency as well as protoporphyrin IX accumulation. It's 11:52, and I could end here, or I could go through the next part of my talk fairly quickly, because the next part is like, it's more preliminary, but I also think it's more interesting. Diane?
How many more slides do you have, Yvette?
About 10.
Okay, it'll be pretty tight. We need to end. Why don't you go ahead and try?
Okay. So the next part of my talk addresses the question on how does iron facilitate terminal erythropoiesis, because there are several points at which this can happen. Up to the orthochromatic erythroblast stage, red cells undergo cell cycle, and DNA polymerase is an iron sulfur cluster protein. Also in some of our data, we found that iron and heme deficiency can also hinder nuclear condensation and enucleation. So in fact, in the beginning of my postdoc, as I was working on proteins such as TMEM14C that are required for heme synthesis, I also found that concurrent with heme deficiency, The TMEM14C gene trap erythroid cells also had a nuclear condensation defect, and they didn't even, make it close to the enucleation stage. And in a more recent study, in a knockdown of FAM210B, which was a protein that we found to be required for optimal mitochondrial iron uptake during terminal erythropoiesis, there was a defect in cell proliferation as well as erythroid enucleation. So we wanted to know why this was so. We were familiar. I mean, when we talk to people in the iron and erythropoiesis field, like there were, I felt that a lot of the answers to this were well, there's a differentiation defect, so of course this is going to be a problem. But I felt that it was kind of a hand-wavy explanation that didn't quite address the question: how does iron promote these processes of terminal erythropoiesis? So to do this, we decided to use the zebrafish system, and because pretty much we wanted a system that we could manipulate iron status in the whole animal, either genetically or pharmacologically, and be able to visualize red cell development. Because the analogous experiment in mice would be to do an in utero iron transfusion, which is really not straightforward to do. In fish, you just add the drug to the fish water. I think we all know why zebrafish is a wonderful experimental model. We were aided by the existence of a LCR globin fish line, where red cells are marked in green, so you can see them beautifully circulating. So as always, before we could tackle the problem with zebrafish, there were some pretty major technical hurdles to overcome. Firstly, we wanted to sort red cells from single zebrafish embryos and be able to genotype. And right up to when we started this project, this was really not feasible in the fish field, because fish embryos are tiny and you lose a lot of cells in the facts. So most people genotype by phenotype, meaning that you in-cross two heterozygote fish and then you sort the anemic embryos, which should be 1/4 of your clutch. And then you fax those in bulk, and then you fax the normal looking fish in bulk. So these would be your het and wild type, which is an issue if your gene of interest has a phenotype when haploinsufficient. And of course there are also issues with rigor. So we worked on circumventing the technical limitations of sorting single zebrafish embryos, which were largely solved by fixing the fish embryos prior to sorting. So as part of our earlier experiments, we sorted these LCR GFP zebrafish erythroid cells, and we observed that, like mammalian red cells, their morphology changes during development, and they do undergo nuclear condensation, and their nuclear to cytoplasmic ratio decreases with time. And we also found that the intensity of the benzidine stain, that's the heme stain, which correlates with differentiation, inversely correlates with nuclear size. So the more differentiated the cells, the more condensed the nucleus. So we then worked out the system and looked at the mitoferrin 1 mutant fish that were crossed into globin LCR GFP. So as a reminder, mitoferrin 1 is a mitochondrial iron transporter that is localized to the mitochondrial inner membrane and is required for supplying iron for heme synthesis. And we found that consistent with the mouse data, the mitoferrin 1 mutants had decreased numbers of red cells if you look at the intermediate cell mass. And we also confirmed this by FACS analysis. We then made sure that we could accurately sort and genotype single mitoferrin mutant embryos. So we sorted these erythroid cells onto slides, and these are the genotyping results from Sanger sequencing. So you can see that the mutant erythroid cells have really severe morphological defects, and their nuclei do not condense. So we then asked, what can we rescue by iron supplementation? So if the problem is just iron supply to the red cells, we should be able to deal with all the issues of mitoferrin deficiency by giving these embryos a bunch of iron henochidiol. So on the left, you see these cells which we sorted from wild type and mitoferrin 1 mutant fish embryos. And we found that the red cells in the mutant embryos are hemoglobin deficient. And then we treated a parallel clutch with iron henochidiol. So here we do see that there is a rescue of hemoglobinization in individual cells, but if you look at the yolk blood region, you don't get restoration of red cell numbers, and we had also confirmed this by FACS. So our next question was, can you rescue the morphology? And we did this by looking at them by Gamsa staining. So the mitoferrin 1 mutant erythroid cells have really large nuclei that take up almost the whole cell, and you can barely see the cytoplasm compared to the wild type cells, which have a border of cytoplasm. And treatment with iron does not do anything to rescue the morphological defects, although it certainly rescues hemoglobinization. So to summarize, exogenous iron supplementation can rescue hemoglobinization, but not erythroid cell number or cell morphology in mitoferrin 1 mutant zebrafish. And also that iron supplementation is necessary but not sufficient to rescue severe anemia from defects in mitochondrial iron transport. So this leads us to our hypothesis that maybe in addition to needing iron, you need the presence of intact transporters such as mitoferrin 1 and other transporters to form these structural highways maybe across organelles for iron trafficking. And this line of thought is supported by evidence from Reddy's group that iron is trafficked from the inner mitochondrial membrane to the nucleus more rapidly than the cytosol or other organelles, and this process requires mitochondrial remodeling proteins. And one of the key questions in the field is how is iron and heme trafficked to the nucleus, and we are considering the possibility that maybe there are these protein-protein interactions between transporters, iron transporters in the mitochondrial nucleus, and then maybe it gets like assembled into other protein complexes, but we don't know. We are currently working on this in our lab, and we know that this is an active area of exploration in Amit Reddy's lab also. But I think our take-home message is that iron supplementation is not sufficient to reverse erythropoietic defects caused by genetic defects in iron transporters such as mitoferrin 1. So we do need to keep digging. So I want to end by thanking the people who contributed to this work. Firstly, I want to thank my lab members for setting up my lab and getting all of these experiments working. The ClipX patients were seen at INSERM by Hervé and Gael, and Sarah Ducombe, who is now a postdoc at Boston Children's, did most of the ClipX genetics. John Phillips and his team at the Center of Iron and Heme Disorders did all the iron porphyrin quantitation and enzyme activity work. And lastly, we are very grateful for our funding sources. And I am now happy to take more questions if there are any.
Thanks, Yvette. Any questions? If you have them, please just chime in, unmute yourselves and ask.
Hi, this is Stefano Rivella. Thank you so much. It was a very nice presentation. I really enjoyed. So I want to go back to the data that you presented before on the relationship between iron administration and rescuing the phenotype in male cells and also in the patients. I think it's a very fascinating story. So obviously in male cells, I don't think there is any relationship between male cells and hepcidin because you're just providing the iron to the tissue culture. So can you elaborate a little bit more on the possibility that not enough iron gets to the cells? assuming that TFR1 is expressed at the correct level, or if there is maybe a deficit in iron intake. So assuming that TFR1, so in other words, assuming that TFR1 does work as expected,
yep.
You need more iron. Can you maybe elaborate a little bit more, just for me, why you need more iron inside the cells, higher concentration of iron to overcome the defect in having more ALAS2?
Yes. So I think what we found was that the heme defect was actually caused, we think, by a combination of PPOX activity, FEC activity, as well as iron utilization. When we looked at mitochondrial iron content, there was actually no defect in mitochondrial iron content, but adding additional iron was able to partially rescue the phenotype. So this is kind of an intriguing question. And our guess right now is that there might be some issue with maybe chaperoning or shunting iron towards heme synthesis rather than iron sulfur clusters, because we did check iron sulfur clusters. We checked SDHB activity and we checked aconitase activity, and both were normal. So right now we think that there is a problem in bringing iron into the heme synthesis complex, although it's there. So, if you dump a whole bunch of extra iron onto it, like maybe there's just like extra iron to go there. But at this point, we don't know how this is happening.
So then not necessarily is the stabilization of ALAS2 the problem, right? Maybe ALAS2
No, it's not. No, I don't think ALAS2 activity is the problem at all.
Oh, I see. Okay, that was what I was trying to understand. So
Yeah.
so some higher level of ALAS2 may not be the cause, maybe just a side effect of the defect in this model. Okay.
Yes.
And so, so the other question I have, so I noticed that the patient that you were, the data in the patient, they definitely had a very low level of transferrin saturation and serum iron.
Yes.
So clearly,
yes.
So it looks like this somehow maybe this defect also leading to a defect in iron absorption. I wonder if you can speculate. You have any idea about that?
So likely not, because the uncle and the patient, the proband's uncle and father, which also have the same ClipX mutation. If you look here, their serum iron is pretty much normal. Their transferrin saturation is pretty much normal. So I don't think the ClipX mutation directly caused the patient's iron deficiency. I think that's likely some other problem.
Oh, I see. Okay, I was trying to understand. Okay. But it's good that they basically providing extra iron. And this was oral iron?
Yes, this was oral iron.
Okay.
Yep.
Okay, thank you.
Yep.
Any other questions?
Just to get a final clarification, does ClipX have any role in PLP binding to ALAS?
In yeast, likely yes. In vertebrates, we haven't formally checked, but likely no. Since ClipX deficiency doesn't, the ALAS is fully active in ClipX deficient cells.
Thanks.
Yep.
Okay, we're gonna cut it off there, and I want to thank Yvette for her talk today. The recording will be available on the CIHD website, and thanks for everybody's participation. And our next speaker will be Kevin Hicks on January 26th, who's going to talk about a new technology that's being developed here at the University of Utah, discovering metabolite binding to specific proteins and how it regulates protein activity. And so I hope you'll all be able to join us for that talk, because it's a really interesting technology that is being developed here at the University of Utah CIHD. And thanks again, Yvette, for your presentation.
Thank you, Diane.
Okay, talk to you later.
See
Thank
you.
you.
Bye, everybody.
Thank you. Bye.